Hello everyone, I am Suchita Bharambe, Assistant Professor, Department of Microbiology, Government Institute of Science. I am here to present my seminar in online refresher course in Biological Sciences conducted by UGC HRDC Dr. Babasaheb Ambedkar, Marathwada University. So my seminar topic is enzymes. Welcome everyone. Today we will explore the fascinating world of enzymes, the biological catalyst that play crucial role in increasing rate of chemical reaction within the cells. Enzymes are the incredible molecules that operate without undergoing any overall change in themselves. They increases the rate of reaction without involving, without suffering themselves. So let's dive into the world of enzymes and understand their significance. Enzymes are primarily the thermolabile organic catalyst synthesized by living cells. One of the remarkable features of enzyme is their ability to function even in absence of living cells. Enzymes can be either proteins or RNA and possess highly specific catalytic activity, that is, they could target the specific substrates only. The history of enzyme says that term enzyme was coined by Wilhelm Kuhn in 1878. It originates from the Greek word en means in and zyme means yeast. In essence, the enzyme were initially associated with yeast. Over time, scientists made significant discoveries like isolation of diastase, amylase in 1883. Buchner in 1897 showed fermentation takes place in the yeast cell extract without yeast cells. And the crystallization of urease by James Sumner from jack bean extract, it is reported in 1926. So these are the glimpses of history of enzymes. We will discuss the structure of enzymes. Enzymes composed of two parts, mainly the inactive protein and non-protein component. The inactive protein, it is called as apoenzyme, which becomes active upon binding with the non-protein part, a cofactor, ultimately forming a holoenzyme. So these cofactors, they could be the coenzymes or the prosthetic group. The coenzymes like NAD, FAD, CoA are the non-protein molecules which are loosely associated with the enzymes. On the other hand, prosthetic groups were firmly attached to the protein and difficult to remove from the protein. The prosthetic groups like heme, biotin, flavin, iron sulfide, coppers, they could work as a cofactor. Types of enzymes. Enzymes can be classified based on their location and their regulation. Extracellular enzymes are secreted by cells and their functions outside the cell. While the intracellular enzymes, they were synthesized inside the cells and they carried out their catalytic activity inside the cell itself. Whereas The constitutive enzymes, they were produced in the cell at a constant rate, whether their substrate is present or not. While the inducible enzymes increases their production when their substrates are present. So first we are going to discuss the extracellular enzyme. Extracellular enzymes, the exoenzyme is an enzyme that is secreted by cell and function outside the cell. Say the digestive enzymes in spider, flies. They are the best examples of extracellular enzymes which help for the extracellular digestion in the spiders, flies. Exoenzymes, they were again used as a virulence factor to cause the diseases. Exoenzymes play a crucial role in pathogenicity of certain bacteria and fungus. They are used as primary mechanism to cause disease by allowing the invader to break down the host defensive outer layer and penetrate deeper into the tissues of host to set the infection. For example, coagulase facilitates blood clotting while hyaluronidase help pathogens to penetrate inside the tissues. Let's explore some specific examples of exoenzyme and their functions as virulence factor. Coagulase, produced by Staphylococcus aureus, facilitated clotting of blood, protecting the bacterium from the host defense mechanism. Thus, it facilitated invasion of bacteria inside the host. It acts as host defensive mechanism, defense against the immune system of host. Kinase, also from Staphylococcus aureus, dissolve clot, helping in bacterium survival. Another example, like staphylokinase, it allows to dissolve the clots. Exoenzyme could work as a digestive enzyme. Apart from their role in pathogenicity, exoenzymes are also vital in digestion. Digestive enzymes like amylases, pectinases, and lipoprotein lipase catalyzes the hydrolysis of various substances to help in nutrient absorption and metabolism. Next type is intracellular enzymes. Intracellular enzyme is the enzyme that produced and functions inside the cell. The best examples are the enzymes involved in the metabolic pathways, the glycolytic enzymes. These intracellular enzymes are essential for proper functioning of the cell and overall metabolism of the cell. Another type of enzyme is constitutive enzyme. Constitutive enzymes are present in constant amount inside the cell regardless of the cell's metabolic state. These constitutive enzymes, they are actually doing the function of housekeeping and are vital for maintaining basic cellular processes. For example, enzymes of glycolysis enzymes or TCA cycle are the best example of constitutive enzymes. Inducible enzymes, in contrast to constitutive enzymes, are present in very small amount inside the cell initially. But they increases in concentration when their substrates are available. A classic example of inducible enzyme is beta galactosidase, which induced by the substrate lactose. Again, these enzymes could be exhibit different structural arrangement. They can be monomeric consisting of single polypeptide chain, or they could be oligomeric composed of multiple polypeptide chains. Some of the enzymes form multiple enzyme complexes where catalytic domains occur on more than one polypeptide chain. The best example of multimeric enzyme is pyruvate dehydrogenase complex, which play an important role in cellular respiration by converting pyruvate in series of 5 reaction to acetyl-CoA. Actually, pyruvate dehydrogenase, it is not a single enzyme. It is complex of 3 enzymes along with 5 cofactors. The 3 enzymes are pyruvate dehydrogenase, dihydrolipoyl transacetylase, and third is dihydrolipoyl dehydrogenase. Along with these 3 enzymes, this pyruvate dehydrogenase complex includes 5 coenzymes like thymine pyrophosphate, CoASH, lipoic acid, FAD, and NAD. So the conversion of pyruvate into acetyl-CoA, it is carried out by this multi-enzyme complex. If we are going to analyze the characteristics of enzyme, enzymes are protein or RNA. They are thermolabile in nature. Enzymes are genetically determined and they have large globular structure with unique 3-dimensional shape. Enzymes accelerate reaction without altering themselves, without altering chemical equilibrium. Enzymes are required in very small quantity to carry out the chemical reaction. Enzymes could be precipitated by organic solvents like ethanol, inorganic salts, for example, ammonium sulfate. Certain enzymes exhibit reversibility of the action. Also, enzymes have very high catalytic efficiency, high degree of specificity, but they were sensitive to their environment, the physical environment like temperature, the chemical environment, change in pH, change in metal ion concentration. Catalytic activity of the enzymes could be regulated by small ions which could functions as a cofactor. So this is the basic about enzymes.